Right. All right, can you guys hear me well? Yeah, okay. So we're going backwards. As you noticed yesterday, we had like advances in tools and spike sorting. We mentioned spike sorting a lot. Then Pierre presented spike sorting. He mentioned SpikeInterface a lot. And now I'm presenting SpikeInterface. So I'm Alessio Buccino. I'm an electrophysiology pipeline engineer at the Allen Institute for Neural Dynamics and lead neuro data scientist at Catalyst Neuro. And I will talk about like an overview of SpikeInterface. Again, we're going to have like hands-on training in the afternoon, but at least you know what we're talking about. So Pierre introduced these extracellular signals. I will actually go quite quick on these slides because if you're here, it means you know what we are talking about. But I just want to stress the fact that over the course of the years, we had so many different ways of recording extracellular neural activity. So people have been using tetrode and are still using tetrodes for many applications. So these are bundles of microwires that you insert in the brain and you can actually reach a deeper region during the experiments. You have silicon probes, so you have electrodes embedded on silicon substrates and provides like additional spatial density to your recordings. And then the CMOS technologies started being integrated into these probes. And we had like planar high density microelectroarrays like Maxwell Biosystems and Tree Brain, which have like thousands of electrodes that can be recorded simultaneously. And for in vivo, we've seen yesterday and today as well, we have neuropixels and synapse probes that basically bring a whole new level of spatial density for recording extracellular activity. But all of these recordings need to go through the same very essential processing step, which is how do I get spike times out of my recordings? And these are very essential for many applications that I would argue that in most of applications for systems neuroscience, say you're looking at place cells or visual receptive fields, whenever you need a receptive field, you need basically very accurate spike sorting. There are some spike sorting less applications like looking at manifolds or brain-machine interfaces, but this doesn't mean that we can forget about spike sorting. Spike sorting will always be an essential step in systems neuroscience. Hopefully, otherwise we're all out of our jobs, right? When I started with my PhD in 2006, I did some experiments myself. So I implanted tetrodes in rats in freely behaving environments. And then I got my raw data and I was like, finally, I can crank out some spike sorters. And I was like, okay, where should I start? There was actually in 2016, there was Clusta and Spike in Circus was developed. And then Kilosort came by, JR class, Wave Cluster, and so on. And I think many of you, if you've been in the field in that period, you felt a little bit lost. I mean, at least, what should I do? I mean, could I try this and that? The problem is that it's not so easy to try different sorts. At least it was not so easy at the time, right? So you sit there, you look at the documentation for a cluster, you see, oh, ouch, my data format is not correct. I need to convert it. What about the parameters? I need to tweak this one and this one. I need to create a parameter file and so on. It was very tricky to set up different spike sorting pipelines. And especially maybe if you are in a lab with different acquisition systems, they come with different file formats, right? So already like the first part, so injecting or ingesting your data in the conversion in the pipeline is a first complication. And especially in electrophysiology, differently than other fields, we have like a lot of different acquisition systems and all of them come with different formats. So we need a first parsing effort. And then we start looking into the spike sorter. So we figure out the conversion. And we see that there are many different technologies underlying the different sorters. Some of them are in Python. Some of them are accelerated in C. Some of them require a GPU. Some of them are in MATLAB. So you need definitely a layer in order to be able to properly interface with these different sorters. And all of this means that in order to actually run a spike sorter, you need to sit there, make some custom code to interface your acquisition system and data to a specific spike sorter and go on with the analysis. And this is not really good for science. It means that a PhD student that maybe starting with this project, they need to spend a lot of time and effort, and maybe they haven't been trained exactly for this like heavy programming and tweaking, and so they might make some mistakes. And all of this hinders reproducibility, right? So if you ask me, how did I do my first Spike story in 2016? I would tell you, oh, I have no idea. And I couldn't even find where my code was and so on. So we tried basically to come together as a group, and actually the last part of the talk will basically cover the history of this project and how we all got here. But the idea was to create a framework, which is in this case a Python package, to make everything easier. So the first and foremost goal of SpikeInterface was to be able to run multiple Spike sorters on the same data set with minimal effort, right? And then basically we produced something. We started bundling additional functionalities for pre and post processing, computing quality metrics, doing curation. So all of the things that Pierre mentioned in the previous talk. We started also, since now we had like a standardized framework to run multiple sorters, we start comparing them, right? What's the best sorter out there? Like how these two sorters, do they give the similar results? Which one is better than the other? We started embedding like powerful data visualization. And then we tried to actually make all this framework reproducible. So using version control, using basically all these software engineering tools and good practices. So 10 years from now, I could say how exactly I process my data set and reproduce it. So just to give you like a visual understanding of what we attempted to do, was trying to go from this manual workflow, which is what I described before. So you have your, let's say, OpenIFIS acquisition system. And so you have an OpenIFIS folder there. You need to write some code to read that data, to parse all these data sets into a format that a Spike sorter of your choice, let's say, mountain sort likes. And then you need to create parameters files for that sorter to run it. And then your downstream analysis will also be bound to the choice of your Spike sorter, because, for example, mountain sort will produce a certain set of files, and then you base your downstream analysis off of those files. Instead, we took like an abstraction approach, and we tried to abstract away the way that we read from the raw and the sorted data. So with this standardized interface, right, now we can forget about where our data came from, and we represent it in a SpikeInterface as the same object, as a recording, right? So once you read the data, right, of course, the reader is specific to a certain file format, but then you can literally forget where the data came from. And once we make this interface standard, we can also easily wrap all the Spike sorters that are available, because we know how this recording object behaves, right? Again, we can forget about where the data came from. And we can do the same thing with the sorting output. So the data that the sorter produces, instead of like reading it directly, we have a thin layer on top, and we basically standardize the way we access the data that we need from all those different files. And since we have this standardized interface, we can now also build tools that make use of this interface to bundle additional functionalities. And now with SpikeInterface, we actually have a whole set of tools. We will also see a lot of those in the demo from later, and also in the afternoon. We have a bunch of pre and post processing tools. You can compute additional features from your data, compute quality metrics, make very fancy and cool visualizations. And this is all thanks to this standardized interface. We can also very easily build pipelines. Again, we know that how this recording object behaves, so we can make a pre-processing steps that apply some pre-processing and also is a recording itself. So we can easily build these like processing chains prior to Spike sorting. And then we get a sorting object, and we can do the same way for like curation. For example, we can merge units or select some good units and reject others. And all of these are represented as the same object. So this really facilitates this pipelining capability. And now actually the framework has grown a lot. So we support, I would say probably everything that's available out there in terms of like acquisition systems, Spike sorters, and Spike sorting outputs. So it's over like 25 file formats. If you don't see your acquisition system here, reach out to us. We'd be very happy to try to like extend it, because that's the goal, to be able to be a unified framework in the field. All of the available Spike sorters, at least the main ones that have been published and are used by the fields, are supported, and you can run them with one line of code, actually. And we also have a bunch of Spike sorting output that we can read from. You can see that we have more Spike sorting formats than Spike sorters, and this is because some acquisition systems, while you record, they might also do some online Spike detection or online Spike sorting, and we can read from those as well. So now SpikeInterface is like a large Python package. It's subdivided in modules, and I will very briefly go through them. So you have also an idea for later and for the afternoon. We'll cover different aspects of this like now large and like complex package. So we have a core module which provides this like abstraction layer that I mentioned before. So it defines the base objects that are used throughout the entire, all the other modules. The extractor modules is the one that provides the I/O functionality. So it has all the data readers. The pre-processing allows you to do all kinds of like processing prior to Spike sorting, such as filtering, denoising, normalization, and so on. Then the sorters modules as wrappers to allow to launch all the different sorters that are available out there. And with the post-processing module, we combine the recording and the sorting object, and we compute additional features like waveforms, templates, PCA scores, spike amplitudes, and you name it. The quality metrics allow you to compute a bunch of quality metrics that can be used to assess the quality or the goodness of your spec sorting output, also hopefully automatically or at least semi-automatically. The comparison module here is the one that basically allows us to compare the different spec sorting results or compare a spec sorting result with some ground truth information for benchmarking. Then we can export to different like third-party software. The widgets module provides all sorts of visualizations. And then we have a couple of like work in progress modules that are meant for automatic curation and cleaning of the spec sorting results. And sorting components is what Pierre presented yesterday. So breaking down all the available spec sorters into individual components and be able to mix and match and compose your own spec sorting pipe. So you've seen these like, it's big, but again, by the end of the day, hopefully you're going to have it all figured out and we're here to help. But before we start, let me give you like these three main core concepts and objects in SpikeInterface. So the first that I mentioned before as well is a recording object. So the recording object is the base object that interfaces with the extracellular raw traces, right? So it allows you to retrieve all the relevant information that we need for spike sorting and for post-processing and visualization and so on, such as what are the channel IDs, what are the sampling frequencies, what are the channel locations, and this is very important for like high density probes. And of course, it allows us to retrieve the traces, but on demand. So you would say, I just want the traces from 1 second to 2 seconds on this subset of channels. And these are basically grabbed from the file lazily and on demand. And then these objects can be easily manipulated. So you can concatenate them, split them, you can aggregate channels or select a subset of channels and so on. The sorting object is the same as the recording, but for spike sorted data, right? In this case, the relevant information is, of course, getting the unit IDs, the sampling frequency, and the spike trains, so the spike times given by the spike sorters. Finally, the sorting analyzer is a third and quite recent, I will cover this later, object that basically is the core of all the processing that we can do in SpikeInterface, and it basically combines a recording object with a sorting object, right? And these two information are essential if you want, for example, to extract waveforms, right? Or compute the average extracellular template of a unit, if you want to compute PCA, compute similarity scores across units, and so on and so forth. So what can we do with all this, right? Let me give you a very direct example. Let's say I sit in my lab, I make a recording, I use OpenIFIS with some tetrarods or whatever probe you like. So the first thing I need to do, of course, is read the recording from the OpenIFIS folder, right? And then maybe if I use tetrarods, it's very convenient to tell the spike sorters that, you have separate probes, right? Every tetrarod is kind of far away from another one, so it should be spike sorted separately. So we can do this by, we can basically inject this information into the pipeline by setting a probe object. Then we're gonna do some filtering, like a bandpass filter. We're gonna do some denoising, for example, removing the common noise with a common median re-referencing. And then we can run any spike sorter that we like. Let's be like modern, and let's run kilosort 4, right? Then we create this analyzer object, and with this analyzer object, we can do post-processing and compute additional metrics. And finally, if you want to do some curation, we could export to file, right? So if 8 years ago, so when I started my PhD, you asked me, can you do this for me? I would say, okay, sure, see you in 1 year. Now, luckily, you can do this in SpikeInterface, this 8 steps pipeline in 8 lines of code, right? And the API is very high level, very declarative. And this is because the main target for this package is experimentalists, right? That basically they do the recording, they work in a lab, and it should be easy to get these things, this standard analysis done. So you can see that the first step was reading OpenIFIS. We call the read OpenIFIS function from the from the extractors module. Then we set a probe file to inject the probe information. We use the pre-processing module to apply a bandpass filter and a common reference in chain. Then we run kilosort 4, 1 line of code. We create this sorting analyzer object that allows us to compute additional information. In this case, we compute the templates and the quality metrics. Again, more details coming up later. And then we can export everything to Phi for manual curation and inspection. And of course, then we can read back the data from Phi as well. Okay, so this was basically what SpikeInterface allows you to do. And now I'm just going to present what was the initial effort that basically motivated this project, which was basically to try to find a structure and understand, like, across all different spike sorters, is there a lion in this jungle, right? So what is the best sorter out there? And in order to do so, we actually use the SpikeInterface comparison module. And let me very briefly explain how spike sorting comparison works. So we have like 2 kinds of analysis. So in one case, we know the ground truth. For example, if we use the simulated data, right? So if we use simulated data, then we know exactly when each neuron in the recording is firing an action potential. So we have this exhaustive ground truth. And what we do with this basically is comparing the spike trains of each ground truth neuron to each spike sorted neurons, and we get an agreement score. So how many spikes were matched, right? Once you get this agreement score for every possible pair of ground truth and spike sorted units, right? You get this like agreement matrix, and then you can do an assignment task. So you can say, okay, what are the actual neurons that are matched? Once you have matched these like pairs of neurons, we can compute all sorts of performance metrics. For example, in this case, we can see that, I don't know, ground truth unit 8 was matched to sorted unit 6, but the match was partial. It was only like, I cannot read it very well, let's say a 60% accuracy. So we can compute all these performance metrics. And we can also further classify units based on this performance metric as being like well-detected units, bad units, like units found by the sorter, but not in the ground truth set, or if it was an oversplit or an overmerge or a redundant unit, we can basically give a label to each spike sorted unit. We can also compare multiple spike sorting outputs, right? And the idea the initial idea is the same. So we can perform pairwise comparison between the outputs of the different spike sorters, right? And then we build a graph where each node is a unit found by an individual spike sorter, and the edge is the agreement to every unit found by other spike sorters, right? Once we build this graph, we can interrogate it and ask things like, give me all the units that were found by at least 2 spike sorters, right? Or give me how many units were found by 50% of the sorters that we run, this kind of things. So we actually applied these like concepts to first real data. So this was the first and main SpikeInterface paper in 2020. We took some data from the Allen Brain Observatory and we ran 6 of the, like at the time, available spike sorters that were actually developed for high density probes. So these are herding spikes, KiloSort 2, Iron Plus, 3D Clue, SpikeInCircus, and HD Sort. And long story short, there was very low agreement in the outputs. So all in all, more than 1,500 units were found by these 6 sorters, and only 33 were in agreement across all sorters. So the main message was like, let's be careful because, spike sorters disagree with each other. And then what we try to do is trying to understand, like, if we could use this agreement to help us spike sort our data. So we turned to simulated data. So we simulated NeuroPixels recording with this MEAREC software. So we tried to replicate the features that we see like in real data, but now we know everything about the spike activity. So we know when each neuron in the recording is firing an action potential. So then what we can do is compute performance metrics. So in this case, it seemed that KiloSort was performing best, but KiloSort was also finding a lot of bad units here labeled as false positives. We could also replicate to a certain extent what we saw in real data. So there was also a level of disagreement here. Of course, a little bit, there was a higher agreement because this data is definitely simpler than the real case scenario. But then we also noticed that most of the units that were false positives, so only found in the sorter and not in the ground truth, were not agreed upon by any pair of spike sorters, right? So basically, with this information, we suggested we could use an ensemble approach to spike sorting, so combining the outputs of multiple spike sorters to automatically clean up the spike sorting results. And this also brought up to this twin project that also Pierre mentioned, which is Spike Forest. So we applied the same comparison concepts at large scale. And this project had more than 1.3 terabytes of ground truth data, running many different sorters with a nice website where you can visualize the results. I don't think it's dead. I think it will revive. So we are in the process to try to update these with more novel spike sorters and ground truth data, and to provide the users some guidance to say, oh, if I have this kind of data, maybe I should use this sorter because it performs better than this other one. But definitely when this was out, it pushed the spike sorting developers to improve their tools, right? Because they wanted to show off as being better on this platform. Okay, next, I will cover some other features that hopefully will make your life easier when it comes to analyzing extracellular electrophysiology. And the first thing is basically dealing with the probe. So in my first slide, I showed that there are many different types of probes out there, and we have to provide this information into a spike sorting pipeline because spike sorters will use this to actually improve their performance and to make use of the spatial organization of the channels. So we developed a project which is called SpikeInterface. And SpikeInterface, basically, it's a framework that allows you either to create from scratch a probe and parse it and pass it to SpikeInterface. We also have a probe public library that now contains all the probes from Cambridge Neurotech, which is one of these vendors, and we're happy to include more probes and more options as they become available and if they want to collaborate with us. And then you can also automatically put a wiring or do the mapping between the electrodes and the actual acquisition system. So just to give an example, here's basically we have Cambridge Neurotech probe. It's called AC116E1. And then this is usually recorded through an Inton chip. And then basically, based on the way the probe is connected to the chip, the different contacts get wired differently on your trade. Yeah, Andrew.
How does this framework deal with programmable probes that might have the same overall layout of all the contact, but can have some that are active differentially?
Like NeuroPixels?
Yeah.
Yeah, for NeuroPixels, actually, since all the switchable configurations are stored in the metadata, the probe object is automatically loaded. So it basically picks the right channels and configuration based on, say, the XML OpenIFIS file or the meta file in SpikeGLX. Yeah.
So it's build it in two different ways, right? So
yeah, exactly. So basically what we provide is some automated pathways. So in this case, for example, we have the wiring that is recommended for this probe is using basically a connector, which is called SE116. So using that connector with Inton chip will give you this automated mapping, right? Then of course, sometimes you have to define your own mapping, and it's essential to get things right? Because you need the traces to correspond specifically to a certain contact to maintain this spatial structure. But if this mapping is supported, then getting the probe from this library and wiring it to a device is literally 2 lines of code with probe interface. We'll have some hands-on in the afternoon.
So I think it's because you can't actually flip it backwards, you're supposed to flip it direct. If you look at the assumption, you should put it in. Like, look at the instructions, right there, even though it is bidimensional. I mean, technically it would, yeah. These are completely, he's saying like, oh, if you put the actual head stage on backwards.
Yeah.
Which this would not automatically map on backwards, if you need to put it on the point.
Yeah, no, I think you need to put it on the correct way, yeah. But we can't control that. So the next thing that I mentioned also at the beginning was very hard is to install these different software, right? They come with different technologies. Some of them are in MATLAB, C++. They might have conflicting dependencies as well. So it would be very challenging to have many, if not all, different sorters installed in the same environment. So what we did was basically building containers for each of these Spike sorters. So containers are pieces of software that package the software itself and all its dependencies in an isolated environment, right? And they can run on your machine that acts as a host, right? In a sort of a subsystem with all these dependencies. An example of these containers are Docker and Singularity or Obtainer. So we built images for all these sorters, which are publicly available. And one cool thing that I think we did was basically some of these Spike sorters are MATLAB based. So we actually compiled them. So you can run them even if you don't have MATLAB installed. So say, for example, you want to run KiloSort 2.5, but you don't have it on your machine and you don't have MATLAB. Then you can just say, I want to run KiloSort 2.5, but run it in Docker. And it will automatically start the Docker container on your machine, map all the inputs and outputs, run KiloSort in the container, and give you back the results. That's it.
So one of the problems that was trying to get any with cheap table acceleration.
Yeah.
Because not getting suited to work.
Yeah.
With this low physical. Do you deal with that at all? This
I have to say it magically works. So basically this will map your GPUs and it has all the, so the container has all the NVIDIA like and CUDA libraries installed and it works. I don't know how, honestly.
Values doesn't work with the rerun at IBM that we did on quite a lot of GPUs. And the driver version in the GPU generation has a lot with the latest version of Torch, for example. And this is something that we will have to deal with in SpikeInterface. It's not trivial.
Yeah.
So the Docker thing, we push back the problem back to the Docker NVIDIA container.
Yeah, probably we need to make images with different drivers. Yeah. But I mean, these were built with relatively recent drivers, like say from 2021 to 23, and most of the times it works. But then maybe with the
idea like the 2018, 2019, not compatible anymore.
noted. And yeah, again, I have to mention, I mentioned this later as well. This was funded by the Salmons Foundation, and also it's a project with Catalyst Neural. Another project is the big data problem. So as Pierre mentioned, we are scaling up. So we have NeurPixels, we have Synapse probes, they produce, we have in vitro, we have Maxwell, Treebrain, they produce hundreds of gigabytes of data per hour, and things will get worse or better, depending on like the way you see it. But so we already have NeurPixels 1, for example, generates 80 gigabytes of data per hour. And in vitro, we even like have some prototypes that record 20,000 electrodes simultaneously, and these produce 1.4 terabytes per hour, right? This is a lot. So one thing we implemented is like a compression framework. And the compression framework in SpikeInterface uses ZAR. So ZAR is a very powerful framework for storing chunked and compressed multidimensional data sets. And we actually analyzed different compression strategies for electrophysiology data, like over like 15 compression methods. And it turns out that audio compression works really well for electrophysiology. I mean, not surprisingly, mainly because audio is kind of similar in terms of like sampling rate, frequency content, correlation across channels, and so on. But actually, a few days ago, Neuralink came out with this compression challenge, and just tell them, hey, we have a paper. We should, yeah. But we can compress like NeurPixels 1 down to like 25 to 27% of its original size losslessly with this like audio codecs. For NeurPixels 2, it's a bit worse, but this is because NeurPixels 2 actually has ADCs with like more bits, so it actually stores more information, right? But actually with a few lines of code, you can go from, say, OpenEFIS recording, any SpikeInterface recording, you can define a ZAR-compatible compressor and save the recording to ZAR, applying that compression in parallel. Yeah, I saw Andrew taking a picture. I came back. Pierre already covered this, so I'll go very quickly and just show you how to do it in SpikeInterface. We're going to have also a hands-on in the afternoon. Drift is a problem, okay, we know. The templates change, so we need to correct for it. So now we have this like modularized framework in SpikeInterface that allowed us to benchmark different steps of this complex motion correction machinery, and actually found out what is the one that seems to work best for this kind of data. But what I want to highlight is that if you have your like shank probe, so you need to have to cover a lot of like depth in order for this to work. But then you can just use the pre-processing module and apply the correct motion function. You can give some arguments as well, and we're going to see more of this in the afternoon. And this gives back a recording which is already reinterpolated, so it should not be motion should be corrected for. Yeah, this basically just a shout out to the quality metrics module. So in the quality metrics module, we have over 15 quality metrics that we can compute, and they will give us some numbers on the quality of spike sorting and of individual units. So for example, as Pierre mentioned, we can compute ISI violations ratio, which tells us if you have contamination in the spike trains. You can compute signal to noise ratio, amplitude cutoff, and these kind of metrics to tell if a unit is complete. And then based on these quality metrics, you could actually design an automated recipe that you can also report in your paper, for example, to say, like the IBL is doing, we only kept the units, which has like ISI violation ratio below a certain threshold, an SNR above another threshold, and an amplitude cutoff below a certain threshold. So this is basically an automated and unbiased way to get like a higher quality spike sorting output that you can also report transparently in your paper. Finally, I will mention a few of the visualization tools that we have developed and we see this afternoon. One, it works in Jupyter notebooks. So it allows you to visualize basically any object in SpikeInterface in an interactive way with these widgets. For example, here you're seeing some traces, you can scale them, you can move through time, you can slice the channels you want to see from, change the window size, all of these interactively from a Jupyter notebook. We have a GUI developed by Sam, which is amazing. It's the SpikeInterface GUI. It tightly integrates with the SpikeInterface object model. So it's working as like a visualization tool for spike sorting output. And the plan is to integrate also some curation functionalities so that Phi can retire in peace. And then we have developed with Jeremy Magland and Jeff Souls from the Flatiron Institute some web-based visualization. Okay, I won't show them now because we'll cover them in the afternoon, but basically, oh, let's do it actually. Basically, these are shareable links that are produced, and they're basically, when you launch this plotting function, they upload the data to a public cloud bucket, and then this web app automatically reads the data from the cloud and parses them and visualize them. So this produces these shareable links that when we share the slides, you're very welcome to click on those. You'll see exactly what I'm seeing. And for example, at the Allen Institute, we're using these to check the quality of our recordings, also with different layers. So here we're seeing the high pass data. Here's this common median reference data. And this is the high pass spatial filter that Olivier talked about yesterday. And we can also use these to check the quality of the spike sorting outputs. So here is basically a sort of a web-based version of Phi that allows you to scroll through units, look at the waveforms, parallelograms, and so on and so forth, and also do some manual curation. For example, here it's unit 0. We see the waveform here. You can also say, I want to flag these as noise and so on, and or merge units and then export the curation as an output. Yes. And finally, we have a full compatibility with the NWB format or Neural Data Without Borders that Luis introduced yesterday. So this is like a standard for electrophysiology, and we can read and also write to NWB with SpikeInterface. And we can also interface with this Dandy archive, so this open repository for sharing neurophysiology data. We can actually read a recording from Dandy and just stream them without the need to download them locally. So you could actually inspect the files and play with them without the need to download them. So give me another 5 minutes. I just want to like, instead of the classic, thanks to all the collaborators and like users, I wanted to like explain a little bit the history of this project, right? Because now it's been a while, so it was nice to look back and see how we got here and how now this has become like a mature project that people hopefully can happily use and solve some problems. So it all started in October 2018, actually a little bit before, but October 2018 was the first commit to the SpikeInterface repo by Cole. I don't know if it's here, it was here yesterday. And this came up with a little bit randomly, but we got some, common connections. We got together, and these are the founding fathers. So Matthias, Cole, me, and Jeremy Magland from the Flatiron Institute. And soon after, Sam Garcia over there, he made his first commit. So actually it was like 4 months after, February 7, 2019. He had very, very cool ideas. He had been in the field for a while and so on. So the smartest idea was like to include him in the founding fathers. And then from there, actually, we started working again. The first effort was in unifying and benchmarking the spike sorting framework. So we had an event right here 5 years ago in June. So basically, it's almost a 5-year anniversary called Spike Sorting and Reproducibility for Next Generation Electrophysiology. Here's the 4 of us because Matthias was taking a picture, I guess. And then we actually started laying out the ideas to produce this package and put together a first preprint, also with the help of Josh Siegel from the Allen Institute for all the quality metrics part. This was published in October 2019 on bioRxiv, and finally accepted to eLife in November 2020. And now we were at versions of SpikeInterface 0.9.1. Then for most of academic project, this would have been the end, right? Because PhD students, they graduate, they move on and so on. We were kind of lucky in this good position where Sam is like a research engineer in France. I was also very involved in the project, and we knew that we just did not even 20% of the work. I mean, we just got what we needed for a publication. But then the next phase was making this really useful for the community. So we knew there were some things we had to refactor, and we had to actually develop to make this a useful software that any new PhD student or postdoc coming into electrophysiology could use out of the box. So we kind of were the appointed government at the time. And we made a major breaking change in July 2021. Sam and I mainly Sam, refactored the entire code base. So initially, SpikeInterface was made of 9 separate Python packages, and maintenance was a hell of a nightmare. I mean, it was to make a release, it took us 3 weeks to check that all the versions meshed and so on. So it was very complicated. So we unified all the code into a single repo, which is the SpikeInterface repo. We introduced new concept, like the concept of segments. We integrated it with Neo. We developed probe interface. And the next part, so from, let's say, 2021 to 2023, there was a real push on development. So all of the features that I showed in the previous slides were basically developed during this period. We invested a lot of time, and many people in the community invested a lot of time and helped out. We now have over 60 contributors from the community. And here's just to highlight a few. So we did the compression framework, was mainly me at the Allen Institute, the motion correction with collaboration with the Paninsky Lab, the sorting components framework, spike sorting containers. So we started adding a lot of useful features to make this more useful for the community. And we also had a hackathon in New York City 2 years ago that actually also strengthened this community and collaboration effort. And now we are at December 2023. So we are version 0.100.x, let's say. Sorry, with 0, then you also have a few bug fixes. So I think now we are 0.6, right? And then, I mean, there was a lot of engagement by the community, and many people actively contribute, and they're actually expert in this project. And I think the very first time that this became a community-driven effort to make a standardized package, it started in, when is it? December 1st, we made a first enhancement proposal. So we wanted to make a major breaking change that we thought would actually make the whole ecosystem better in the long run. And many people are now contributing to this discussion, and we actually took us over a month to properly figure out how we should do this and what we should change and what this new infrastructure should look like. And finally, after 1 month of discussion, we made a pull request. We implemented this new sorting analyzer object that you'll see more about in the afternoon. And this will be released probably a couple of weeks from now, so we can integrate things from the hackathon as well. And hopefully this will be the last major breaking change in the API. Luis.
Well, it's like 32.1.4. I started in the.
So there are still a few things we want to iron out. But again, I want to stress this. I think also discussing with the parliament, hopefully this will be the last very big change because now we're happy with the overall flow and, you know, we prepared the package also for integration with third-party packages and like web applications and so on. So we hope maybe even before, but I think sometime early 2025, we can target our 1.0. We need to increase proper coverage, testing, documentation. Yeah, and so if you want to join the community, it would be very helpful if you can actually start the project on GitHub. It seems like sort of like, you know, advertisement on our side. But I have to tell that it's very tricky to apply for grants for this kind of like maintenance projects, right? And they ask, oh, how many users do you have? Yeah, I don't know. We can look at downloads and so on. So actually, these stars on GitHub is a readout of how many people are appreciating your effort, right? And you can actually report this when you apply for this grant. So if you haven't done so already, you can go to the SpikeInterface GitHub repo. You have a star here and you can hit star. So now I'll wait 3 minutes. And if we don't have over 10 stars, I won't, I'm kidding. You can do it, you have until the end of the day. Otherwise, we lock the doors. Yeah, and that's all. Now we're gonna have a break and then a live demo with Sam and with some showing the SpikeInterface and then ends on in the afternoon. Thank you all for the attention and enjoy your coffee now. Questions? Maria.
So SpikeInterface always be tied to. Will SpikeInterface always be tied to one version of Python, or have you designed the coding to be future upward compatible to future versions of Python?
So we actually did a discussion ongoing within the parliament, right? Within the people that are actively maintainers of the code. So how we should deal with this lifetime of different packages. So actually the Python versions, they have a longer support, right? Than for example, NumPy version. So NumPy is the main package for like numerical operations and all the scientific libraries rely on this NumPy. So I think we decided to support all Python versions depending on the NumPy lifespan of the different like versions. So let's say if NumPy stops supporting Python 3.8 from the next release, we should follow along because we heavily rely on this NumPy, and NumPy is really like the foundation of scientific computing in Python. So, but now, as of now, we support all major Python versions, so from 3.8 to 3.12. Yeah.
One more question. The output of the extractors going into the sorters, is that fixed in the sense, is that law interleaved binary format for all the sorters, for some of them, or what? How do you handle that?
Yeah, so some sorters require the input to be like a binary format. So for example, kilosort, not kilosort 4, kilosort 4 actually has a thin wrapper that is, you can pass directly a SpikeInterface object. So we're using that, so you're not copying the data. But for example, for kilosort up to kilosort 3, if you don't have a binary data to start with, then the first step of the spike sorting is to convert and save a binary dot file that kilosort can actually read. So it's a duplication, it's a temporary duplication of the data, and this dot file optionally can be removed. I say optionally because if you then use phi, then phi can read this dot file, so you can check the spikes, the spike sorted spikes.
Okay.
Okay, so let's have a break. We meet back at 11:20. Yeah.
But my boss likes to be great, you were, but. No?
Yeah, because brain also is right.
No, exactly.
So, but I checking it out.
I don't know. Internet is very good for sure. That's sometimes it's good off, but it's. My boss. Yeah, my boss. That's what boss. That's amazing.
And I see there are some elements that just expressed by another.
Yeah, but
I am very nice.
Because it's more of I don't say this in this way. I don't need
My next. Now, we'll see, Evelyn, right, you street right, so.
Case,
Some of them are. I'm sorry. Thanks.
Yeah.
I think it was missing because basically, because you know, the recording to use this, because you don't have the links.
Yeah, I think Alessio added a way of having a base with the lines.
Right.
Okay.
I didn't see it. Recorded, recrossed.
You miss the question?
I can make it.
I think you can start with that. Once people come back.
Let me read.
Yeah, you have knowledge, it's you.
Some things the recording, so that. So you need to